Hello, everyone. My name's Alejandro Chavez, and I'll tell you about our work today, performing high-throughput genomic insertions and its application towards protein tagging. We call our new method HITAG, and we believe it represents a rapid way to probe protein function at library scales. So why do you want to tag proteins at their endogenous locus? One, it lets you study proteins at physiologic levels. And once you do the tagging, you can now monitor things such as protein localization in vivo inside of cells and see where the protein is in real time. Can also identify interactors of that protein of interest, see who it's binding to, see where it's interacting with. And you can control the stability of that protein of interest to understand what its function is within the cell. And so while there's many ways of doing gene tagging, most of them suffer from being only one gene at a time can be targeted. And that's a bit of a problem because there's 20,000 protein coding genes in the human proteome. And protein behavior is known to be cell type dependent. And so if we want to scale protein-wide analysis, current methods are quite inadequate because they require a lot of time and a whole lot of resources to tag everything in the cell. And so what were the overall goals of our project? So develop some sort of high throughput approach to gene tagging. We have a starting cell line. We do some sort of manipulation to the cell line, and we end up with an output that is a library of cells where every cell has a different proteins tagged with our marker of interest. So what mechanism do we use for tagging? Our approach is based on a previous publication called CRISP-Paint. And so in CRISP-Paint, what you do is you take your target gene of interest. In this case, in this example, has 4 exons. We want to tag the C-terminus of this gene. We then have a donor plasmid that has, in this case, our tag of interest, GFP. And downstream of the GFP has a pyrimycin resistance marker. Now what we do is we deliver Cas9 along with 2 guide RNAs. One guide RNA cuts the C-terminus, our target gene of interest. Another guide RNA cuts a donor plasmid. Now that the cell has 2 double-strand breaks, at some rate, they'll fuse together. And if they fuse in frame, then you've now GFP tagged your protein of interest. And if your protein of interest is properly GFP tagged, they'll also express this pyromycin resistance marker, which allows you to select for properly tagged cells by simply growing them in puromycin drug selection.
Hello, I am Joonwon Kim, a postdoctoral researcher in the Chavez lab at UCSD. From here, I will take over and explain the experimental design and results. So both CRISP-Paint and our new method HITAG are capable of generating C-terminal tag cell lines. The key advancement in HITAG is that in a single experiment, it generates a pool of cells where each cell in the pool has a different protein C-terminally tagged. So now, instead of doing multiple rounds of CRISP-Paint, we can do one HITAG experiment. To achieve this library scale tagging, we modify CRISP-Paint into a 2-step process. First, we deliver a set of target guide RNAs against the genes we wish to tag using a low MOI lentiviral infection. This allows each cell to receive one unique target guide RNA that will guide what gene each cell will tag. Second, we transfect the remaining components necessary for tagging. This step initiates the tagging process where now each cell attempts to insert our tag at the C-terminus of a different gene. Similar to CRISP-Paint, properly tagged cells are then enriched by selecting for pyromycin resistance within the population. At the end of the HITAG process, we obtain a diverse pool of tag cells where each cell expresses a target guide RNA and has the corresponding gene C-terminally tagged. Another changes in HITAG is applying dual Cas9 system. While CRISP-Paint use only SP-Cas9, we use SP-Cas9 to cut the target gene and introduce SA-Cas9 to cut donor plasmid. This approach offers 2 major advantages. First, it prevents competition between target guide RNA and donor guide RNA for Cas9 complex formation, maintaining high target cutting efficiency regardless of SA-Cas9 guide RNA presence. Second, it solves a potential recutting problem. In some cases, the tag gene sequence near the PAM regions can resemble the donor plasmid, allowing repeated cutting by the target guide RNA even after successful tagging, which prevents properly tagged cells from surviving. By using SA-Cas9 with its different PAM recognition site, we eliminated target guide RNA accessibility to the properly tagged constructs. In our initial experiments using HITAG, we observed an uneven distribution in tagging among the final generated cell pool. This is shown in the graph on the left, where the size of each box indicates the proportion of each target guide RNA or tagged genes in the population. What we observed is that before tagging, the target guide RNAs within the pool are equally distributed, but after tagging, only a few target guide RNAs are highly abundant, suggesting that only a few genes were efficiently tagged within the population. To address this issue, we further optimized our transfection condition and improved the tag constructs. For example, we increased the copy number of the promycin resistance gene from 1 to 3. We did this because the expression of promycin resistance markers are driven by the native promoter of whatever gene we are tagging. So we hypothesized that some tag genes might be lost in the resistant marker where expression was too low for cells to survive the drug selection process. And increasing the copy number of drug marker will rescue those genes to pass through the final library. When we applied the improved transfection conditions and the optimized tagging constructs, we observed a clear improvement in the distribution of tag genes, with more genes being present in the final tag library with overall list skewness. While HITAG enabled us to tag hundreds of proteins at a time, not all genes we wanted to tag ended up in the final library. When we dug into the data, we noticed that tagged genes had a higher basal level of expression than untagged genes, suggesting that expression of the promycin resistant marker may still be a limitation. In addition, we noticed that tagged genes had a higher predicted efficiency for their target guide RNA as compared to untagged genes. Next, our team used HITAG to gain insights into stress granule biology. Stress granules are transient RNA-protein granules that are known to form during times of translational stress and are thought to play a role in neurodegeneration, though the exact mechanism remains unclear. At the time we initiated the study, several publications had documented hundreds of stress granule-associated proteins. But which proteins strongly accumulated within stress granules and what protein properties mediated this strong accumulation remained unknown. To answer these questions, we use HITAG to generate cell lines with mCherry fused to the C-terminus of over 100 stress granule-associated proteins and track whether the protein accumulates in stress granule under stress condition. Through HITAG, we successfully generated a library of 167 cell lines, each with a unique stress granule protein mCherry tagged. We then took all the lines, induced stress granule formation, and quantified how much each protein accumulated into stress granules by looking at their overlap with the canonical stress granule marker G3BP1. To our surprise, we found that only a small subset of the proteins were found to robustly accumulated in stress granule. When we looked at the features driving strong accumulation in stress granules, it was associated with RNA binding capacity and high predicted propensity to the liquid-liquid phase separation. This aligns with known stress granule biology, where RNA-protein and protein-protein interactions are essential for their formation. So although 167 genes were all reported as stress granule-associated proteins, our studies showed that only a portion of 23 proteins strongly accumulate within this structure, suggesting that association accumulation can be quite different. As shown here, HITAG is a powerful tool for tagging large number of proteins to understand their essential roles in the cell. By tagging all proteins with the same tag at the endogenous locus, it enables a uniform downstream analysis of all lines and allows proteins to be studied at their physiologic levels of expression.
We want to improve the efficiency of HITAG. While it's now working at scales of hundreds of proteins at a time, there's still some bias in our libraries, which we'd like to improve to make it more even distribution of every single protein equally tagged. We also want to adapt the HITAG approach to other cell types. Right now, we've done it mostly in cancer cell lines. Love to expand it to things such as iPS cells. We also want to combine HITAG with other single-cell omics methods, things such as single-cell RNA sequencing or even pooled optical screening type technologies. And we think this will really enable the scaled interrogation of protein function because now you tag in a pool and you study this population of proteins in a pool. And finally, we are also interested in vivo applications of HITAGs, doing tagging inside of animals to study protein function in native tissues. And we actually were lucky enough to receive an NHEJ opportunity fund to help support this research. Thank you.